Okay, so in that we were discussing about viral, non-viral and naked DNA vectors. In that we have discussed about different types of the vectors. What is the function of vector? That is to the that are used to deliver the DNA. Means in gene therapy, what we do, we want to correct the we want to cure the disease by removing the defective gene or by correcting the defective gene. So in this procedure, we need to deliver the DNA. So to deliver the DNA, naked DNA, then vector is used. So these are the different types of vectors. I am late class. Chaliye, class is going on. I will talk to you later. Hmm. So, liposomes, lipoplexes, and polyplexes, these are the non-viral vector system. So, as you all of you know that virus infect us like the corona or any virus enter in our body. So, it induce the means our immune system work against them. Okay, so what is the structure of virus? In the virus structure, either we have the DNA or RNA. Okay, so any foreign DNA is delivered in our body. Okay, the same case is with the vaccine. What is the vaccine? That are also the foreign antigen that has been introduced in our body to induce our immunity. Okay, so viral vectors are used to deliver the DNA into the physical body of the patient. So there are the different types of the structures that can be used. So virus is used. So other than viruses, we can also use the different methods that are liposomes, lipoplexes, and polyplexes. Okay, so you already know the mechanism of the viral infection. So as the virus infect our body, so that DNA also what that is multiplied in our body. Okay, whenever we fall sick. So liposomes and lipoplexes are the other. These are the safe method. Liposomes, lipoplexes, and polyplexes. They have the lipid layer and DNA is introducing into these liposomes. Actually, liposomes and lipoplexes, these are produced by the endosome, by the flip-flop mechanism, okay, in our body. So, these are used as a vector to deliver the DNA. So, the desired DNA that we want to introduce into the patient body. So mutation has a great impact. Means we have started from the SNPs and other variations that play an important role in the prediction of disease, in the manifestation of the disease. So this we can also interact by the RNA interference method. In that the RNA pair with that gene that doesn't allow the other RNA means the miRNA pair with the RNA, it doesn't allow the expression of the mRNA. Okay. So expression of the mRNA is interrupted. So that mechanism is called RNA interference. So this is also called the gene silencing method. Okay. How the gene is expressed? First, mRNA is produced from the gene. During the transcription, mRNA is produced. So, if we want to if we want to inhibit the expression of mRNA, then we need the another RNA to pair with that. So, miRNA is that kind of RNA. miRNA is short miRNA that pair with the. So, this mechanism is called, this process called RNA interference. Sometimes we also induce this RNA interference. Although in the plants or in some of the organisms, it is found actually. So Nowadays, it is in use, and is to cure the cancer, the RNA interference method is used. So this is the important technology, RNA interference, that can make the gene silent, that can inhibit the gene to being expressed. Means no mRNA will not be expressed. So in that way, there will be no disease, no cancer. Okay. So RNA interference is one method. So now we were just finishing that. Today we'll discuss some few resources that are important. We can search on these resources. There is huge information that is available. Lot of information. You can learn a lot. Okay. You can just search about the gene, about which you are interested. So bioinformatics application play an important role in the analysis of the disease, in the diagnosis of the disease, or in any research, in any study. So you can go on, you can search in these databases. So although there are many more, but few I have selected few. Okay, so you can go to the NCBI and search. The first one resource is genetics home references. So we can get everything. This will give you all the information related to human and a genetics home reference provide useful information in the form of all the genes. Okay, gene and gene product which are involved in disease means what is the product of gene means protein. Protein is responsible for disease. Gene orthologs क्या है? The same gene from the different organism, that is orthologs. I have told you in the class, same gene from the different organism, that are called gene orthologs. Okay, who is there? So same gene from different organism, that is called orthologs. Orthologs means homologs. You also call this homologs. Like alpha globin from human and alpha globin from mouse. These are orthology. Genomic regions. So orthologies means if we want to search about the same gene in the different organ, we can study the expression of the same gene in the different organ. So we can have the phenotypic information. What is the function of the gene in different organism? The function is the same, but you can study. You can take the reference. Genomic regions. What is this? That is the functional part. Means that is involved in the how much DNA is involved in the production of protein. That is that genomic region. And transcript of that region, what is the transcript of that genomic region? That is mRNA. Because mRNA is produced from coding DNA. What is genomic regions? Means that codes for the RNA. That is transcribed into RNA. So RNA is the product of transcription. So we use this term transcript, mRNA transcript. SNPs, single nucleotide polymorphism, organism and mutation. So all these information you will get on this database. Genetics home references. You just go and search there about anything, about any SNP, the disease that we have discussed here. You can search about this DMD gene or any gene. You can search. Okay. I may have shown here something. Okay. So you will get lot of information about this genetic information, gene ortholog, genomic regions, transcript means in how many ways the gene can be transcribed, the same DNA can be transcribed. SNP, single nucleotide polymorphism. Yeah. So there are the different disease that is there that is associated with the SNPs. So SNPs have so many manifestations, different kind of disorder, organelle mutation. So you can find out what is the location of the gene, okay, to which phenotypic features it is associated. So you can find out all the details and about the biological process. Organism also, in the different organisms we can look, okay, what may be the biological process. So everything you will get. So lot of bibliographical information, textual information you can get here. This is the important resource. Another resource is gene database. That is, gene database provide information about function of gene integrated from. So information is integrated from other, means we have cross reference on this page, on this database. Reference sequences, maps, means how many sequences are related to this maps. On the map we can identify the location of the gene, so we'll have that information. If we want to correct a gene, defective gene, then we need to know about the location of the gene, then only we can correct, we can remove that gene or we can repair that gene. So this information is important, mapping. In the above database genetic home reference, we have the textual information about biological process or everything. We can read about the disease something. So in the gene we have the specific information related to the gene location of gene pathways, specific information about the pathways. What is the means, you can also get the information about the function of the gene, how the product of the gene is playing role in the pathways. So there may be the product protein product of this the genes that participate in the pathways, different pathways. So there may be the different pathways that may be associated with the disease, or there may be the abnormal expression of the gene in that pathway, that protein is not playing well, protein may be inactive in the pathway or showing the less activity, or so showing the may show the high activity. So disease may be the manifestation of that. So the pathways is responsible. In the biochemistry at biochemical level, we used to, the medicines are given generally to modulate the pathways. Whatever medicine we take, it modulates our pathways, means our other pathways that are affected by that. So it cures the disease at that. All the medicine that we take, okay, by oral or injection mode, so that's only work against the proteins. So it inhibits the expression of the proteins. Sometimes the drug minimize the expression of the protein. Whatever there is a problem, we just design the medicine against that. So that information is important if we want to cure the disease, because we are talking about the gene therapy, okay, or okay. So this information is important in means in the medical, in the diagnosis of the disease, also in the treatment. This information is helpful. Variations. So variations are also important because variations are responsible. Variation within the same genes are responsible for different types of phenotypic manifestation. Phenotypic variation also. So Mendel also described about the variations. So variation has an important role. So in the gene database, we will have the information about the variations also. SNPs is also one kind of variation, and there may be the other type of variations. So variation may be on the appearance. In the first lecture also, we have discussed a lot about the variations. Phenotypes. Phenotypes is the manifestation of these variations at the genetic level. First, the variation was found on genetic level. So Mendel told about that there is something that is responsible for variation. Okay, in the characters, in the traits of the same character like dwarfness and tallness, color, different color, yellow and green. Okay, so there should be the gene for that color. Then only that color will be like expressed. So at that time, Mendel was not aware if there is any DNA, but there is something, there is some element that is responsible. So phenotypes are the manifestation of those variations, and that may be related to the disease. So, and which genome is related to that, which phenotype is related to that variation. So all you will get in this database. So this database is very useful, and you can search. We have searched in this database. In practical, we have seen the practical class. I have told you about this. We have searched about the genes, open reading frame, everything. Okay, we have predicted open reading frames. So many things. So everything you will get here about the gene. And this another important resource that is OMIM, Online Mendelian Inheritance in Man. You should say that human, but name is like that. Online Mendelian Inheritance in Man. The abnormal traits listed in all the genetic disorder. All the genetic disorders have been listed in this database. Mutation, mapping, gene, phenotypic, allelic variants. So you can search in this database by clicking on this link. So abnormal traits are associated with the disease, generally like alkaptonuria. Alkaptonuria, the disease when the urine turns black after exposure to the air. So this is the genetic disorder that is found in the man. Means urine turns black color after exposure to air. So that is one type of genetic disorder that is found in man. So that we can search in the OMIM. So there are different types of genetic disorders that is associated with the abnormal genes if there is a mutation in the gene. That's all we can search. Allelic variants of the same allele, like allele of tall, allele of height, will have two two alleles. Means one for dwarfness, one for means recessive and dominant. So that is, what are these variants? So we'll get all the details on this. Okay, we will search on this database. Right? DB This is another database. I miss our all the previous lecture are based on this. Okay, DBSNP. DBSNP means database on SNP, single nucleotide polymorphism. So you can search about all the types of single nucleotide polymorphism or any polymorphism in this database. Insertion, deletion, microsatellite, and non-polymorphic variants. Means which are not polymorphic. Means multiple variants are not there. So there can be SNP, insertion, deletion, microsatellite. So about all these, we can search on this database. So we have already discussed about the DBS, about the SNPs and polymorphisms in the last classes. So now I will not go into the details. Unigene. Unigene is the another database. Analysis expression by tissues, age, and health status, and provide the information about this. So in the Unigene, this means we can get expression is important for the disease. We are always interested in which tissue it is expressed, what is the expression, how tissues are getting affected, age, health status, and provide the information about this. So expression of gene is important. It affects the tissues level. I mean, whenever tissues are affected, we feel pain or feel good. Or if our body is working, body tissues are working, we feel good. If our tissues are not working, we don't feel good. So tissues are the functional unit of the cells. What is tissue? That is a functional. So whether it is working, whether the system is working or not, it depends on the tissues. So different cells, the group of cells work together. Like the protein works in the group. In the same way, tissues also work in the group. So it depends on the age, also on the health status. Okay, so we can get all the information about this on this database. So the age is also important. At the different age, we can have the different types of different status of the tissues. Okay, and gene expression can also may vary because means as we grow older, the repair mechanism becomes slow. Okay, we need the more nutrients, we need the good food, and as we become older, we can't take the junk food, or we can't. Our body will not metabolize that. Means in the young age, it may happen that you are eating everything and you are getting metabolized. But the manifestation, I mean, these things you cannot eat if you cross 40 or something. Okay, then you need to change your habits because your tissues work like that. Whatever you feed to them, their function will be affected, their structure will be affected, and the function also. If you give the tissue the good food, they will work well. So you can get this information, the health status on the UniG. Another important resource is GEO. This is about the expression also, gene expression profile. You can search for the expression of if you are interested in any specific gene, because any specific gene is responsible for any specific disease. So we are always interested. Okay. Gene expression. Okay. Okay. Contain information about a specific gene profile derived from microarray data. You can also download microarray data from this. What we get here? We get the data in the form of Excel file. In the Excel file, what we get? The expression level, expression of the same gene. The expression means intensity. We will have some another lecture on microarray also, then you will be able to understand. So, in the microarray data analysis, we see the expression of the same gene in different conditions, like the expression of the gene in aerobic condition and expression of the gene in anaerobic. Means what is the expression of same gene in aerobic condition and what is the expression of same gene in anaerobic condition. Then we can compare the expression of the same gene in two conditions. So in two conditions, the gene expression will vary. So these are two conditions, there will be two column. Yeah. So in the Excel form, you will get the data. Gene profile. So that is called gene profile. Single gene can be expressed in the different condition like pH value 1, 2, 3, 4, 5. There may be different expression of the same gene in different condition. So microarray is the technology. Using that technology, you will be able to analyze those conditions. Okay. So important data means that you can, for the practical, you can retrieve the data from here. So if you want to know more, I have the lecture on YouTube. Microarray. What is microarray? You can listen that lecture. Okay. With these slides. I mean, that is also related to this. Okay, I will send you the link of that lecture. Microarray data. Okay. What is microarrays? microarray technology is what. Okay, so I will send you the link of that lecture. Don't worry about that. Okay, so we'll discuss about this first. Means you can. I mean, this is very easy. We can click on the link and we can go. I mean, wherever you want to search. I mean, this is the links are given here. So by clicking on these links, you can search. Like I want to search in this. This should work, I think. I should say whether it is opening or not. This page should open. Why it is not opening, I think. इसको नहीं खोल रहा है. Okay, we'll see कि ये क्या है. In this database you can go. Sorry, ये वेबसाइट है, गलत हो गया थोड़ा सा. We can searching OMI. Okay, that is the same thing. Online Mendelian inheritance in man, an online catalog of human genes and genetic disorder. So here you can see. Like El Capto, I don't know the correct spelling of. Yeah, this is the name of disease. This is the one disorder that I know, alkaptonuria. Means urine turns on black color after exposure to it. So here you can see, you will get a lot of information about. Are you able to see this? All are able to see this? I have typed one name of genetic disorder, alkaptonuria. So now you can cytogenetic location is this 3q13.3. Phenotypic gene relationship you can see here. Alkaptonuria. So if we click on this, we will get the information. Alkaptonuria caused by homozygous or compound heterozygous mutation in homogentisate 1,2 dioxygenase gene HGD. Now chromosome 3q30. So you are getting the detail here. Alkaptonuria is an autosomal recessive metabolic disorder characterized by accumulation of homogentic acid leading to darkened urine pigment. So this is the effect. Urine color becomes dark because of presence of homogentic acid. So pigmentation of connective tissues, ochronosis, joint in spine arthritis, destruction of cardiac valves. So these are the manifestation, okay, of this disease. All are able to see this or not? This is the information that we are retrieving from OMIM database. Alkaptonuria enjoy historic distinction of being one of the first condition in which Mendelian recessive inheritance was proposed. Okay, it was proposed. So this was the first disease in which Mendelian inheritance was proposed. उसका नाम जो database का भी है, and of being one of the 4 conditions in the charter group of inborn errors of metabolism. Okay. सो मेटाबॉलिज्म में कुछ एरर्स हो जाती हैं, उसकी वजह से डिजीज होती है. द मैनिफेस्टेशन आर यूरिन द टर्न डार्क ऑन स्टैंडिंग, एल्कलाइनाइजेशन, ब्लैक ochronotic pigmentation of cartilage and collagenous tissue. So there are many more disorder. So whatever you want to know, you can search. And arthritis, especially characteristics in spine, clinical features. So the different information you can get here. Carrier. Okay. Now we are going back. What do you want to see? Just see. Now, what is this? This is the location. Location is important because location is important for gene therapy. Means how we can use, if we know the location, then only we can correct, we can replace, or we can, okay. If we don't know the location, we will do we are going to damage the patient body or patient body DNA. So we should be careful. Location is important. So this gives the information about location. Alkaptonuria number is this. Okay, mouse symbol related जो भी मॉडल होंगे इससे मॉडल वगैरह that is also given here. Okay, so reference sequence you can retrieve it from here. Draft assemblies for large collection of the genome. Pancreatic cancer के केस का यहाँ पे है genome. एल्कैप्टोनूरिया का ये है. इस पे हम क्लिक अगर करते हैं, so this is the cross reference. So here you can see. Actually on internet we have everything. We just need to look, what is there. So there is a lot of technological advancement. We have so much information on internet. So you just search on the few databases and make a file, practical file. Okay. Assembling, reference assembly, alternative haplotype sequence alignments. Okay. So reference gene sequence is given here from the NCBI, and this is the sequence of allelic variant. Okay. And this is compared with the reference sequence from the NCBI. Okay. So one sequence we have that has the leptonuria disorder, and one is normal. So we can compare haplotype sequence. So in the but specifically in the OMIM we have the sequences that are variants. Here you can see gene expression in 54 tissues from GTX RNA sequence of itne samples. This information we have. Cis regulatory element. What is cis regulatory? Cis regulatory means the two genes, suppose they are inheriting together, they are expressed together in the next generation or in the cell. Means they are same. They are expressing together. एक साथ उनका एक्सप्रेशन हो रहा है, दो एक साथ एक्सप्रेस हो रहे हैं. That are called, sorry, cis regulatory elements are found on the DNA sequences. And there are certain cis regulatory genes also that inherited together. Means दो होते हैं वो. These are two promoters are found out. If the two promoters are found, so two genes will be expressed together. Okay, related to that. So there is lot of information we have. We can click on that and we can enter into the another. Means there is so much information. Okay. If I click on this, I will get another page. So in the detail, we can have this. So here you can see these are the different organisms. Rhesus, mouse, dog, elephant, chicken. So human sequence can be compared with those also. This is alignment actually. Okay, there is a more similarity in the mouse, dog, elephant, and chicken. chick is a chicken and tropical zebrafish. These share some segments, small segments. And you will also get the DBSNP link here means information. Short genetic variant from DBSNP. 153. Okay. So perform this OMIM search on L-captonuria, then save each page. Okay. Whatever you do search, whatever search you are doing, you search, you just save all the pages. Okay. And then make the report of all the practical. You can see, you can zoom this page if you want to see more. There's so many things to see. This is the one example I have taken. Mapping and sequencing. So you can, these are the links related to that. Centromere, GC percentage. These are the different links. Okay. So if you are interested more, you can see. What do you want to see in this? Like fish clone. What is it? Fish method होता है क्या? Fluorescent in situ hybridization. Mapped clones along with the. So the information is given related to that. So this is a filter. Okay. So this can be. So there are the different things that we can use. What we want to do, we want to change color, you can see the different types of things. वहाँ पे height selected था squash or we can choose length and submit. So you can change, you can make change. Yeah. You just go there and click on each link. So here you can see no red. I selected this color no. Tag. Means this is the location. Alkaptonuria gene. Just go in this database and search on them. Okay. So there is the 18 clones. GC percentage you can see. Okay, how many GC content is it there? But generally in the disease in which we are interested, what is the location of the gene that you are able to see. Take a picture of this. This is the location. If you just see, you can see the location. Q13.33 is the location. You can zoom it from here. There are so many other things that we can. So, just leaving this page. So directly you can go to the. I think this will be the same page. This is it. Okay. The captonolia. This is the number. Mouse symbol. Gene phenotype relationship. This is also important. We want to know what is the relationship between gene and phenotype. So here the important information you can get. Captonuria phenotype MIM number is this. Okay, so there are so many variants, manifestation also. You can read about this phenotype, MIM, membrane. So here we can see homogentisic acid oxidase deficiency. There is a lack of this enzyme actually, which is responsible for this disease. So lot of information you are getting. So about the phenotype means the text, it is written in the textual form. Yes, okay. So by clicking on these different links, you can search. What is this? This is the number. In reference to the number, this is the information 264800. Phenotype gene relationship. What is this? Pseudoxanthoma elasticum. Okay. And what is the location? Okay. Gene location is this. Gene locus MIM number is given here. We can see clinical synopsis here and fin gene graphics. Okay. So clinical slab. Shall we click on this? If we are clicking on this, we can see. So here we are getting the outer link also, external link, means clinical trial. So you can get all the information. So these are the phenotype that is associated, means central vision loss, visual impairment हो जाता है, macular degeneration, hyperpigmentation, angioid streaks of retina, retinal hemorrhage, choroidal neovascularization, molecular degeneration. So these are the manifestation of this disease. So all have been written here. Everything has been written here. All the manifestation, all the phenotypes have been written here. Okay, the doctor observed that. Okay, so here we are getting all the information. These are the different types of phenotypic features. So all the information we can get on this platform related to genetic disorder. We can click on another one also. But these are related with each other. Phenotype gene relationship. Now we are going to click on phen gene graphics. What is it? This? So, radial linear. See, this is the good thing. Huh? Now, pseudo xanthoma. Means here you can see in the graphical form how these are related to each other. So these are the different types of, these may be the different types of manifestation, how these are related to each other. So it is shown in the form of tree. Okay. And this one. You can also see it in the view radial means ऊपर से नीचे. Here you can see. So these are related to each other. You need time to see actually. So we are searching in the OMIM database. You can see how much detail we are having. So you have so much information on the internet. So that is all have been obtained by the biotechnological experiment. So on the previous page that you have seen the name of the different technologies. So everything is was there on that platform. Okay. So because we have already done the so much experiment, means the scientists, researchers, they have done the experiments. So by clicking on these also you can go into the details. हम इस पे हम click करते हैं तो we will get more information. Or okay, we can prune back the tree. Okay, if you want to focus on the few things. So I'm just closing. Now this is the phi engine graphics we have done, clinical synopsis we have seen, and we were searching in the OMIM database. Okay, so we have seen the phenotype gene relationship, phenotypic studies we can see, and we have also searched about the location of the gene. This was important, and then we have searched in this. Okay, so this is important. I just want to show you, let me call you. So here, these are the name of different means what we need. Everything is here. So by clicking on this, you can go into the details. CRISPR, this is the new method. CRISPR tag. So CRISPR is a new method of genetic engineering. In using this method, using CRISPR technology, you can do modification at the exact location. While in the traditional genetic engineering method, we cut at the near about position where only restriction enzyme can cut. But using the CRISPR-based method, we can cut at the appropriate position. This is the benefit of the CRISPR technology. So here we have different link PFARM database. Okay, discuss on the. You just do this again and save all these. Okay, I'm just saving one page. Okay, sorry. You do this practical and save this as the PDF. I'm just saving only single page. Okay, I'm saving it here. Okay, do you have any query? Now lecture is over. If you have any query, you can ask. Okay. So in one source we have SARS. Now you have seen there are a lot of information that we were able to see. Okay. B.Sc Biotechnology. Is it your class? Subject computational so now do this practical at your home, then send it to me. Make a practical file also. How many practical we have done? You just make a file and show me. I want to see your file. Details of all the practical in the MS Word. Okay? Just make a now type your attendance in the chat box, then you can leave the class. So in the next week, the VC sir will visit the department. So they will see your report also, practical report. Okay. So practical file you should have. You just type your attendance and leave the class. Do it fast. Have you done your attendance? Now I'm closing. Have you done? Just do it fast. How much time will you take? You do it fast. Type your name and roll number and leave the class. Do it. How many left?
मैम
Shalini, Hanisha, just do it fast. Okay.
मैम ये
Print.
इंफॉर्मेशन रिसोर्सेज जो हैं इन पे प्रैक्टिकली क्वेश्चंस आएंगे या थ्योरी में भी आएंगे?
प्रैक्टिकल भी लिखना है ना, सब आना चाहिए ना इसमें। जब मैंने
ये
क्रैश फाइल बनाई है पहले। देखिए,
यस मैम।
क्वेश्चन कैसा भी आएगा, लॉन्ग क्वेश्चन आएगा, देन यू कैन इंक्लूड योर प्रैक्टिकल आंसर, दैट विल गिव यू मोर मार्क्स, है ना, द एग्जामिनर विल गिव यू मोर मार्क्स।
यस मैम, मतलब हम लॉन्ग आंसर टाइप क्वेश्चन में इंक्लूड करेंगे।
Include, oh, you should include. You
यस।
should you should write down the examples, cite good examples, and practical reports. You should cite there. Okay.
यस मैम,
Then
लाइक
only
पर्टिकुलर क्वेश्चन नहीं आएगा?
it may be. It depends who is going to make the paper. It depends on that. Okay.
यस मैम। यस, थैंक यू।
Hmm? Is type your attendance हो गया? Type your attendance. I'm closing. I'm stopping the record now. No more attendance will be taken now. Okay. I'm just stopping the recording. Then attendance will not go on the record. Okay. No more attendance now. Have you done? Everybody has done or not? If I click on the stop, there will be no recording. Just give your attendance. Now attendance is now over. I'm closing. Done.